Ever had a cell culture ruined by contamination? Or maybe you've wondered if you're doing enough to keep your experiment sterile. In today's video, we're going to cover the essentials of sterile technique in cell culture step by step, from prepping your workspace to handling cells. By the end, you'll know exactly how to avoid contamination every step of the way. Let's start by talking about why sterility matters. You might think it's just about keeping things clean, but it's really about ensuring the integrity of your results. When we culture cells, we're creating a perfect environment for all kinds of organisms to grow: bacteria, fungi, yeast, even tiny contaminants like mycoplasma. And if any of those get into your culture, it can change your results, waste your time, and set you back days or even weeks. So, following sterile techniques protects your experiment and gives you confidence that your data is reliable. Alright, now let's get into what you need to do before you even start the experiment. First, personal preparation. Make sure you're wearing a clean lab coat, gloves, and a hairnet. It might sound obvious, but these basics are your first line of defense against contamination. And avoid wearing any jewelry or makeup that could shed particles into the workspace. Next, let's talk about setting up your workspace. Disinfect your biological safety cabinet with UV light for 30 minutes. Ensure all surfaces are adequately exposed. A cluttered space is just asking for contamination, so keep things organized. Arrange everything you'll need, like pipettes, media bottles, and tools, within easy reach to avoid unnecessary movement. And lastly, preparation of your equipment and reagents. Wipe down all bottles, flasks, and tools with ethanol before they even enter the cabinet. Pre-label any media and solutions too. Trust me, you don't want to realize halfway through that you need to write something down. Now, let's go through each stage of the cell culture process and look at what sterile techniques to use at each step. Step 1. Media preparation. When you're preparing media, the first rule is to keep everything as sterile as possible. Wipe down the media bottles and reagent containers before placing them inside the cabinet. Use fresh, sterile pipette tips for each reagent, so you don't risk cross-contaminating them. And be mindful when opening bottles. Only open them as much as you need and avoid putting caps down on any surfaces. Step 2. Preparing the flask or dish for cell culture. Once you're ready to work with your flask or dish, place it inside the sterile area of the cabinet and avoid putting it right near the edges where contamination risk is higher. If you need to remove a cap or lid, hold it or place it upside down on a sterile surface to keep it clean. Step 3. Adding media to the flask. When you're adding media to your flask, make sure you're using proper pipetting techniques. Avoid touching the sides of the flask with the pipette tip and control the angle and depth carefully so you don't end up splashing media around. And as always, use a fresh tip for each different reagent to avoid cross-contamination. Step 4. Handling cells and adding them to media. Now, when it's time to add your cells to the culture media, use aseptic pipetting. Be extra careful here because this is where even a minor contamination can ruin the entire culture. Handle your cell containers and pipettes carefully and avoid passing your hands directly over open containers. This helps reduce the risk of airborne contamination. Step 5. Incubation and monitoring. When you're ready to incubate, carefully transfer your flask or dish to the incubator. Make sure the incubator is clean and regularly disinfected, especially the internal surfaces. Keep an eye on it during your experiment to ensure nothing unexpected is growing. Step 6. Post-experiment cleanup. And finally, let's talk about cleaning up. Dispose of any waste immediately and appropriately. Don't leave anything sitting around that could become a contamination risk. Wipe down all surfaces in the cabinet with 70% ethanol one last time, and then expose the cabinet to UV light for 30 minutes. As you remove your gloves and lab coat, be careful not to spread any contaminants outside the BSC. So, that's it. Those are the essential steps to keep your cell culture experiment sterile. It might feel like a lot at first, but once you get into the habit of following these steps, it'll become second nature. Stick with these habits, and you'll be able to trust your results and avoid setbacks. Thanks for watching, and I hope these tips help keep your cultures clean and your experiments running smoothly.